Assalamualaikum and greetings to all, especially to my examiner, Madam Fazia, and my supervisor, Dr. Hairul. My name is Fatin Natasha Shaira binti Amar Azwan. So here I'm going to present to you about my final year project entitled Sequence and 3D Structure Analysis of Metroxylon sagu cysteine protease. So here is the abstract of the research, where all the details information will be elaborated more throughout the presentation, which is also involve introduction, literature review, methodology, result and discussion, and finally is a conclusion. Metroxylon sagu, or known as a sagu palm, is a multi-stem true palm tree that belongs to the order Ericalis, family Palmae, and subfamily Columbidae. It grows quickly, is dominated permanently in both lowland freshwater swamp and also have been found in a dry land. It also have a higher in terms of starch productivity compared to the paddy, which have a multitude of use, including crafting and food sources. However, cysteine protease is a thiol protease that play a vital role in the growth and development of plants. It activated in a programmed cell death and senescence, which involve in a pathway of signaling along with the biotic and abiotic stress responses. It also contributes to the degradations and maturations of protein, as well as housekeeping functions for abnormal removal and unfolded protein. However, the discovery of cysteine protease in sago palm is not well studied, including its sequence and structure analysis. A limited research focusing on this protein in sago palm, making a huge gap understanding its function and mechanism evolved. Therefore, the research aimed to understand the sequence and structure by comparing the cysteine protease from Metroxylon sagu with other cysteine protease from plants, animal, fungi, and protists. Firstly is a protein structure prediction, which is necessity for understanding the functions of the protein. There are three main methods, including computer simulation based on energy minimization, knowledge base such as homology modeling and a threading approach, and finally is a hierarchical approach. However, in this research, we are going to use homology modeling, which is the most influential in predicting protein structure that can generate reliable results with a higher accuracy. Secondly is homology modeling via computational analysis, where the software that will be used are NCBI+, which is a sequence similarity search program, CLC sequence Workbench, which is available for multiple alignments. Then we have a Phyre2 and SWISS-MODEL, which both are the software that will be used to design the 3D models of a selected sequence. And finally is SAVES version 6.0, which is applicable for model optimizations and validations to test the model via energy minimization through a series of tests including ERAD, Verify 3D, WhatCheck, and ProCheck. Thirdly is protein domains, MOTIF, and foldings. In a secondary structure, a protein folding such as helix bundles, Roseman fold, and beta barrel will build a small patterns repetition in a protein structure called as MOTIF, which is known as a supersecondary structure with a unique chemical or biological function in a protein structure. However, an accumulation of MOTIF into a larger subunit called as domains, which is a unit of conserved sequence behavior observed via evolutionary related genes comprising genes family. Here we have the methodology, which is include a data selection of 20 sequences generated from plants, animal, fungi, and protists by NCBI+. Then we have the alignment for multiple sequence alignments to observe the conserved sequence and similarities. Then we have a building of homology models designated by a SWISS-MODEL and Phyre2. And finally, we have a validation for optimization and validations of the models by uploading the tentative models in a SAVE version 6.0 in a PDB format. So here is the picture of the conserved domain in predicted Metroxylon sagu cysteine protease by conserved domain database NCBI+ search tool. The three conserved domain known as inhibitor I29 domains, peptidase C1 domain, and GRAN domain also have been found in the cysteine protease from plants, animal, and protists. However, the fungi contain herpes BLF1 domain and peptidase C1 domain. Upon the conserved domain observation, we have found that C1 peptidase domain have been observed in all the sequence selected. Based on the MOTIFS database nomenclature, C1 peptidase family, also known as papain family, which consists of two subfamilies known as papain-like and bleomycin hydrolase. Besides, the amino acid residue known as glutamine, cysteine, histidine, and asparagine have been found as an active site on the conserved domain peptidase C1a, which are responsible for the protein's activities. Cysteine and histidine are catalytic residue of C1 family peptidase for catalytic dye formation. However, glutamine and asparagine plays a vital role in the catalysis process. So here is the picture of the conserved motif generated by the multiple motif exhibitions online software, where all the cysteine proteins selected except for fungi contain at least 3 common motifs. However, each of all cysteine protein sequence from plants, animal, fungi, and protists contain a special motif 1 indicated as red color. The sequence share a same sequence signal such as their active sites for activations of the mature enzyme by the proteomer removal or cleavage mechanism through autocatalytic process activation. Now it's about a protein folding generated by the Phyre2 software. The table presented a various percentages of folding even though the template generated from all 5 queries was the same. It is result from the side chain interactions with their constituent amino acids. So this phenomenon is a requirement to adapt the correct formations of 3D structure mediated by other proteins activity for a proper folding. So let's move on to the multiple sequence alignment. The picture shown as a multiple alignment of 21 selected sequence by COMPUTATIONAL NCBI. The conserved sequence or the similarities between the sequences are indicated as a red bar. Therefore, the analyses have come out with 2 conclusions, which is the first conclusion is cross-species conservation or autologous, which means that the sequence may not undergo evolution. However, the second conclusion is result from the basic stability of reproductions, cellular, and the functions. From this analysis, the functional elements also can be observed in order to help in the medical research, risk of disease assessment, and comparing their amino acid residue at analogous domains and evolutionary histories of distinct species for disease discovery or treatments. Next section is about homology modeling and comparative features of a predicted structure. Upon the 2 software, SWISS-MODEL provided a higher percentages in terms of structure identities compared to Phyre2. Therefore, the table shown a summarization obtained from the SWISS-MODEL data. Based on the table, the Q mean is expected in accurate agreement. However, the individual set score are applicable for scoring functions to indicate which model feature exhibit substantial deviations from the expected native action. Therefore, the Swiss template model library ID 1PCI1A templates was selected as the best predicted 3D structure of Metroxylon sago cysteine protease with 45.91% sequence identity. So here are the structure generated by the Swiss model. And finally is quality assessment and result model validation. The table shows the structure validation by SAVE version 6.0 through series of tests. The chlorophyll primer cysteine protease from protease was the most accurate model. This indicates that the model was passed since at least 80% of the amino acids score higher than 0.2 in their profile of 3D and 1D structure. This statement is also applicable to all selected sequence except for the fungi, which is the Thermosophora pertusa. However, the result from one check, such as bond lengths, bond angles, omega angles of all structures generated will within a normal range, even though there are some errors on the backward conformational due to the unusual protein foldings. However, the Ramachandran plot generated from the project have indicated a good background conformational since all residues are corresponding with non-allowed areas appearing in the Ramachandran plots. As you can see, the residue in the most favored regions is 84.4%. As a conclusion, the predicted 3D structures provide a framework to interpret experimental analysis of cysteine protease and Metroxylon sago for further approaches to explore its functions and mechanisms. Since the results were good and reliable, this could be applicable in a medical research for disease treatment and discovery. So here is the list of references used in the presentation. That's all for me. Thank you for watching.